So what we're going to do now is talk a little bit more about the hormones and how they're moving around and really how they end up working like they do. So hormones, like it says here, can be freely circulating. A good number of them are just freely floating in the blood, not bound to anything. Other ones can be bound to transport proteins. Certain ones it's going to be necessary. So like thyroid hormone, for example, it's bound to carrier proteins. Things that are water soluble tend to dissolve really easily, are easily transported in the blood because it is mainly water. So like parathyroid hormone, for example, dissolves into that really easily, transported around, can diffuse in or out of the bloodstream with this concentration gradient. Lipid soluble ones, again, we know lipid and water doesn't mix, and blood is mostly water. Because of this, we're going to a lot of times need carrier molecules to kind of ferry these things around and be able to get these to travel in the blood. So some of these are going to be very specific to a particular hormone, and some carriers are pretty promiscuous. They can bind a number of different hormones. Like it was the example here, thyroxine binding globulin only binds that thyroxine. Where there's certain other things like albumin, which can bind a number of different hormones that need a transport. So again, some are selective, some are much more promiscuous. Again, these are generally detaching and reattaching type of things. We don't want any carrier protein to bind it up completely, generally, because then it would never be able to detach. So they can be in those two different types of things here. But really what we're trying to do, or not things, but those two different forms, either bound or unbound. What we want these to be able to do is protect these hormones so they're not getting broken down in the bloodstream before they get to their target. And that's also part of the reason even some water-soluble hormones are transported that way, because they do kind of tend to get broken down. Because again, at some level, unless we want to be continually sending a signal after we release something, no matter what the hormone is, it is going to get broken down or removed in some form or fashion from the bloodstream. Otherwise, a single release would be in there indefinitely. So that's why we have some of these transporters even binding stuff that can flow freely, is to let it be present for a little bit longer. And again, there's a number of different things that can influence how much of a particular hormone is in there. Obviously, the big thing is going to be is how much are we making. So more synthesis means more in the blood, which means a higher blood level. But we're going to see these hormones, as I was already saying, are removed from circulation in a number of different ways, much like neurotransmitters are removed from the synapse in a number of different ways. So we are going to have things like hormone elimination through enzymatic breakdown. The kidneys are going to filter a lot of this out. It can be taken back up by certain cells, or taken into target cells. And again, any of these mechanisms of elimination are going to decrease the blood levels, and again, means that once the signal is sent, it's not going to stay out there indefinitely. And we can really measure this based on how long it takes for about half of it to go away. So a lot of times when we're talking about hormones, we can talk about half-life of these hormones. Shorter half-life mean they need to be replaced more often, or they're going to be out of circulation more quickly. Half-life is that time to reduce the concentration by half. A lot of times you hear half-life with radioactivity or isotope changes, but it's the amount of time for about half of this to be removed. Water-soluble proteins tend to have much quicker half-lives. They tend to get into circulation and get removed from circulation really quickly. Where steroid hormones tend to be in there a little bit longer. Some of these lipid-based ones, so for example, testosterone, it's going to take about 12 days for it to go from its starting point to about half of it being there. And then it would take another 12 days to get to a quarter of the amount that was in there. So you can see that it's going to be a little bit longer time course. And again, hormones are going into the bloodstream, and the simple answer of where does blood go in the body, the answer is everywhere. So that means hormones are really going to get sent throughout the body and get to almost all tissues of the body. But only those with the receptors are going to be target cells, because, again, the hormone, I always kind of say this with the endocrine system, is a lot like a radio broadcast. I'm sitting here right now. There is radio broadcast. If I turned on a radio and turned it to a particular station, I could get that broadcast. because I have the correct receptor or receiver to tune that in. It's the same thing with the hormone being in the bloodstream. It's going to almost all cells, but only those that have the right receiver, the receptor, can bind this hormone and get its signal and therefore bring about some type of change in that cell. And again, this is going to be a little bit different in terms of where the receptors are at and how they work depending on the type of hormone it is. So like it says here, receptors for things like those, most of those protein-based ones, the catecholamines like epinephrine, norepinephrine, some of those eicosanoids, those tend to be cell surface receptors. Where thyroid hormone and a lot of the steroid hormones are lipid-based, those can travel through that external membrane pretty easily, and because of that, we end up seeing cytoplasmic or nuclear receptors for this. And again, because of that, how the signaling is going to go downstream from that and their mechanism of action can be a little bit different. So water-soluble proteins, if you remember back to AMP1, we talked about cell signaling. These are doing one of those second messenger systems. So you're having a cell membrane-bound receiver or receptor. It's going to bind to that. It is going to then require machinery on the inside of the cell that connects with that, and then is going to send that signaling pathway. So we talk about these being second messenger systems, meaning the hormone is the first messenger binding to the receptor, and then on the interior there's going to be something that is influenced by the receptor to become the second messenger, which is then going to trigger generally a cascade of events that signal transduction pathway to bringing about some type of action within the cell. A lot of times can be changes in cellular activity. It might be changes in gene production or stuff like that, but that tends to be more steroid-based hormones a little bit more. This is just showing you some examples. Again, I'm not going to ask you to know this thing here, but this is showing you this idea of hormone binder receptor activating a G protein, which can do downstream signaling. Similar thing here, slightly different pathway. Again, similar thing here, hormone binds to this, activates different things that might open up a channel. Again, all these things are cell surface binding, bringing about some type of change in there and bringing about a second messenger. So in the case of this one, you open up the channel, the calcium acts as the second messenger. Inside here, the G protein is acting as the second messenger. I always kind of say protein-based hormones are ones that we're talking about second messenger systems. It's a bad analogy, but it's kind of like smuggling something into prison. You need somebody on the outside and somebody on the inside. That's kind of what's going on with these second messenger systems. You need the receptor on the outside that's connected to the inside to get to the inside there where we can bring about the changes. Generally, the lipid-based hormones are a little bit different because they are lipid-based. They can generally travel through that membrane on their own and usually are going to bind to a receptor somewhere in the cytosol or in the nucleus. And these, for the most part, are going to actually bring about changes in gene transcription. So these generally bind with the chromatin in the nucleus and are going to alter really gene levels, how much we're transcribing something. So mRNA levels of something. You might be turning up a gene, turning down a gene, turning on a gene, turning off a gene. But they're generally affecting that type of thing a lot more with these lipid-based ones. So generally get some type of transcription change in the mRNA leading to more protein synthesis or less, and that a lot of times might alter the cell's structure, might alter the cell's metabolic activity, might cause the cell to make something else, a different hormone, that is then going to lead to the release of this. So like with testosterone, we know it's going to cause certain proteins to be made, leads to more muscle development, changes in the larynx, changes in the hair follicles and how much hair you produce are all responses to this. And this again is showing you that idea of these either cytoplasmic, well that's mitochondrial, but cytoplasmic receptors, nuclear receptors. But again, most of these ones it is going to be some type of change in gene transcription. And because of increasing or decreasing transcription, we might see increasing or decreasing translation of a particular protein. And that can either turn up or turn down some type of cell response, or turn on or turn off a cell response. Again, these water-soluble ones, there's a lot of times multiple responses, and this is true on a lot of these. I mean, even the lipid-based ones too. You can get multiple responses from that hormone there. So, for example, here, certain hormone activation, it might activate or inhibit an enzymatic pathway. It might lead to cell reproduction. It might lead to cellular secretion. It might do multiple ones of these. It might lead to muscle relaxation or contraction. A number of things like there. Main thing is, depending on what that receptor is connected to and what those pathways connect to, you can get a large number of actions from that single signal. So it's not usually one hormone, one action in response to the hormone. A lot of times multiple things can happen with the release of that hormone based on what the receptor, if there's different receptor subtypes, and also what those receptors are connected to. Let's give you the example here of glucagon. So this was when blood calcium gets a little bit too high. Released from pancreatic cells in response to that, goes to liver cells, and then leads to phosphorylation of certain enzymes in those liver cells. And again, if blood glucose is getting too low, it causes these liver cells to release glucose. Showing you the example of oxytocin, how it changes these signaling levels here, leads to muscle contraction. And again, much like we talked about in AMP1, there tends to be these cascade events and amplification of those responses, so we can get a large response from a rather small signal. Again, multi-step pathways usually involved in this. This is all stuff we really talked about at the beginning of AMP1 when we were doing that brief introduction to cell signaling. But it's that idea of that signal transduction and amplification. And again, it's just giving you some examples of things that this might do, these pathway controls, how we can turn them up or turn them down. And again, depending on what we're talking about here, there can be only certain target cells, or stuff can have a lot of target cells here. But any singular target cell, any single cell can be a target for a number of different hormones. They can have different number of receptors for the same hormones, or different amount of, it can have receptors for many different hormones. It really depends on what the cell is doing and what its job is. And again, we can mess with the activity or the receptiveness of this cell, or based on either upregulating or sometimes downregulating the amount of receptors that it has. So we can upregulate, like I said, and downregulate receptor levels, which means these cells can become more, what's the word I'm looking for? Sensitive. They can become more sensitive to really the level of hormones that are in there. Again, more receptors means more sensitivity. Less receptors means less sensitivity to these different things here and changes in those gradations. And this is just kind of showing you this idea of an upregulation or downregulation of receptors. And a lot of times this is going to be part of a feedback mechanism. Is if you're getting a large amounts of this hormone being released and you're getting these big responses, a lot of times, in a lot of cases, you might start down-regulating some of these receptor subtypes to try to blunt that response a little bit. And again, these can be based on where you're at in your development, what the cell cycle stage is in the cell, how active the cell is being at any particular time. These are all things that could be influenced by these receptor fluctuations. And again, generally as these things mature a lot more, they become less responsive to certain types of hormones. They don't need, like for example, growth hormone, don't need to grow at that quite the same rate. And they, like I said, tend to get a little less receptive to that type of stuff. The other thing we can see is there's a lot of times can be multiple hormones targeting the same cell, target cell binding different hormones. And what we can see is we can get some differing effects on this one. Sometimes these hormones can work together. And you can get what's called a synergistic interaction, where the activity of one hormone is reinforcing the activity of another. So estrogen and progesterone acting upon the uterus, for example, it leads to bigger changes in the uterus than either one of those alone. We can also get permissive actions, meaning that one hormone is required for the next one to be functional. So pretty much you need prolactin to produce breast milk before oxytocin can cause release of it. So that would be a permissive action. And then, not surprisingly, we can have antagonistic interactions. A lot of times we see these in these kind of levels of this too high, levels of this too low type of thing here. So, for example, glucagon and insulin act in opposite directions on blood glucose levels. So they would be having an antagonistic effect. And again, that's just a little summary slide, doing it more of a cartoon version, talking about these ideas, but either working together, one needs to happen for the next one to happen as permissive, and then antagonistic is working in opposite directions. And like I said, you can have certain cells that are responding to having many different receptor subtypes that can respond to many different hormones. So what we're going to work on from here on out is then we're going to start taking a little bit more of a look at what is going on with overall how we're regulating the system and the different glands, the hormones they make, and what they do. So that's what we will start with in the next one here. So I will see you next time.